Physiological and molecular plant pathology, and also biotechnology in plant disease management. Now we'll see what will be the probability questions that we can expect from this physiological and molecular plant pathology side for ASRB net 2023 plant pathology. So no, when it comes to physiological and molecular plant pathology, it's for me also it's like a puzzling topic, because many of the concepts are the very deep, and still I couldn't find exact proof how it is actually functioning or something like that. But anyway, I will try to add my knowledge and put my knowledge forward. And my name is Shankar Reddy, and I'm working as assistant professor in Kasar, Best IU. So starts with DNA barcoding. Hope you guys know what is the DNA and RNA, deoxyribonucleic acid and ribonucleic acid, where DNA was first discovered by Watson and Crick in 1952 and 53 at the time, and double helical structure. Now it starts with DNA barcoding. So actually this DNA barcoding was first proposed by Paul Hebert in 2003. So this DNA barcoding is actually used for the identification of species and allocating unknown sequences to higher taxa such as order and phylum. So this barcoding system can be used for the identification of species and identifying or allocating the unknown sequences to the taxa or order or phyla. So rather than telling in detail and all those things, let me tell you what will be the probability questions that we can expect. So the questions will be like, who discovered this DNA barcoding system? Paul Herbert. So what will be the what will be the use of this DNA barcoding system, allocating the unknown sequences of higher taxa to the order or phyla and all those things. And for the species and all those things that we can identify by using this method. And the standardized 500 to 800 BP sequences is mostly used for the identification of this many eukaryotic organisms and all possible taxonomic groups. So here I think this question was asked somewhere. I actually not remembered, maybe in it or somewhere else this question was asked. So what will be the standard base pair length in the DNA barcoding, something like that. I have seen the questions before. So it's a 500 to 800 BP base pairs. So coming to the marker selection, these are all some markers are available in DNA barcoding. And coming to the animals, COI, CYTB, 12S, 16S, so such kind of markers were used. And when it comes to plants, MATK, RBCL, ITS, bacteria, COI, RPOB, 16S, and CPN60 RIF. And for fungi, ITS. So most commonly used marker or locus is ITS, internal transcribed spacer. So RPB1, RPB2, 18S. And when it comes to protists, also ITS, internal transcribed spacer, COI, RBCL, 18S and 28S are the different kinds of markers or locus can be used in the identification of this organisms group. So coming to the one gene, one enzyme hypothesis, I'm not going to explain one enzyme hypothesis, but I would simply say who discovered and who evolved this concept, or who came with the idea of this one gene, one enzyme hypothesis. So before this one gene, one enzyme hypothesis was very famous because it was actually formulated by Beadle and Tatum. Beadle and Tatum come up with an evolved idea of one gene, one enzyme hypothesis, which means each gene responsible for the production of a single enzyme that in turns affect the single step of a metabolic pathway, which means very simple. So every enzyme will be involved in gene responsibility, or every enzyme is a part of the gene responsibility that performs each and every single step in the metabolic pathway. So later this was now it is not in function, because I would say that nowadays scientists are identified many genes are involved in a single functioning, or many genes and enzymes are involved in a single functioning or metabolic pathway of organisms. So this Norman Horowitz named this concept as one gene one enzyme hypothesis. So one gene one enzyme hypothesis in 1948. So before that it was called as genes produce enzymes that control the single metabolic step. Later Norman Horowitz named this concept one gene one enzyme hypothesis, but the concept was given by Beadle and Tatum. Coming to the hypersensitivity and mechanism of hypersensitivity, so it's like a cell defense mechanism or programmed cell death where the cells near the pathogen infection can die or sacrifice themselves to prevent the further infection or further spread of the pathogen. So when it comes to the synonyms or alternate names, suprasensitivity, hypersusceptibility or hyperallergy, these are all the various other terminologies or synonyms can be used for this hypersensitivity. And the concept and the name was first introduced by E.C. Stockman, very famous one, very famous man. E.C. Stockman introduced the concept and the term of hypersensitivity. Hypersensitivity means programmed cell that are autolysis to prevent the further spread of the pathogen infection. And HR occurs only in a specific host pathogen combination. Specific host combination, okay, specific host pathogen combination. So now we will see some elicitor. So the term elicitor was coined by Noel Keane, and I told you this Noel Keane is also considered as father of molecular plant pathology. So elicitor means it is actually a molecule and other stimuli that is induce the synthesis and accumulation of antimicrobial compounds in plants. So this molecule can induce the synthesis and accumulation of antimicrobial compounds, that is phytoelections, and stimulates the plant defense mechanism. So here there are some elicitors and origin, where it is actually originated from and their action has been given. So chitin oligosaccharide originated from fungal cell wall, general defense and initiation, and no cell death. Coming to the flagellin or LG22, bacterial pathogen, initiation of basal defenses. Lipopolysaccharides LPS, bacteria, initiation of basal defenses. Hairpin, bacterial pathogens, initiation of cell death. Cryptogen and Phytophthora cryptogia, initiation of defenses and cell death. Victorine, Cochliobolus or Cochliobolus, initiation of cell and VB genotype of votes. And the last one is NIP1, Rhynchosporium scalis, and the initiation of cell and RS1 genotypes in barley. So coming to the phenolic theory, this is very important theory that was given by J.C. Walker, and we can expect many questions from this. Not many questions, and there are one standard question that was asking repeatedly from many times. I will tell you. So J.C. Walker was first to propose this phenolic theory. Phenolic theory was first proposed by this J.C. Walker. So let me tell you what is phenolic theory. So when it comes to red onions and white onions, there was a disease called onion smudge. So this onion smudge is very severe, or white onions are highly susceptible to this onion smudge when compared to red onions. Red onions are highly resistant because it contains two phenolic compounds called catechol and protocachic acid. So due to the presence of these phenolic compounds, red onions are highly resistant when compared to white onions to the onion smudge disease. So they will ask questions like this. So onion smudge is caused by Colotrotricum cercinans, or what are all the two compounds or phenolic compounds present in the red onion so that it can withstand against the onion smudge disease? Catechol and protocachical. They also ask one question like so what are all the compounds or phenolic compounds are absent in white onion so that it can be highly susceptible to onion smudge. It is something like that they will ask. That is procachical and protocachical. So these are all the very important compounds or phenolic compounds that are present in red onions, and these phenolic compounds are absent in white onions. Phytoelections. So actually phytoelections are the low molecular weight antimicrobial compounds that is produced in the plants during pathogen infection or maybe biotic and abiotic stress. Abiotic means pathogens like parasites, parasites, whatever it is, pathogens or parasites, and like abiotic stress like nutritional disorders or maybe some other kinds of responses. These phytoelections are produced in the plant. So phyto means plant, alexin means protecting substances. These phytoelections are actually plant protecting substances that are produced in the plant. So actually these phytoelections are actually first proposed by Muller and Borger in 1940. So the concept or the phytoelections were first proposed by Muller and Borger in 1940, in Phytophthora infest system, potato and Phytophthora infestans system. So these are all the various kinds of phytoelections that is produced in the host plants. So the first one is rice, Saccharinatin, mobilactin A and B, potato, Rishetin, stilbene, phytoberine and lubimine, sweet potato, Hypomearamon, tomato, Rishetin, Arabidopsis thaliana, camelexin, French bean and bean fasciolin caviton, and tobacco, glutinosin and capsidol. P, pisatin, it's a very important, and it is the first phytoalexin to be discovered by Muller and Borger in 1940. So the phytoalexin produced by P is pisatin. Broadbean, viron and vironic acid, and pepper capsidol, cotton rose, gossipol, soybean glycerolin, red clover trifluororesin, vegetal and macanin, garlic alixin, and broadbean P, viveronic acid, carrot, tosocomarin, alpha medicapin and sativin, chickpea P, cysterin and macain, and parsin root, xanthotoxin, and papaya dial. And these are all different kinds of phytoalexin and the pathogens that was involved in. So like in previous year question papers, maybe they will ask like tobacco phytoalexin, what is the phytoalexin produced by tobacco, or maybe P or something like that they will ask. So coming to the phytoanticipants, so phytoelections and phytoanticipants are more or less similar, but I will tell you what will the main difference. So these are all the low molecular weight compounds, and that is produced before infection of microorganisms. So when it comes to phytoelections that are produced after the plant infection, so examples like saponin, avinacin, tomatin are the examples of this phytoanticipant. So for example, like if you take wheat take disease, that is Gaumonomyces graminis triticea, that produce phytoanticipant called avinacin, which actually degrades the saponin. So the world's first phytoalexin is pisatin, as I told you, pisatin, that is produced from P. So it was first discovered by Muller and Borger in 1940, actually isolated from P. So these are all the pathogenicity related proteins. So there are 17 families of pathogenicity related proteins were identified so far. PR1, 2, 3, 4 and 17 until continuous. So PR1 is tobacco PR1A antifungal, tobacco PR2 beta 1,3 glucanase, tobacco 3 is PQ, chitinase type 1, 2, 4, 5, 6 and 7, and tobacco R chitinase 1 and 2, tobacco S tramatin like, tobacco inhibitor S protease inhibitor, tomato P69 endoproteases, cucumber chitinases, chitin type 3, tobacco lignin formation or peroxidase, and parsley PR1 ribonuclease like tobacco class 4 chitinases. These are all the various kinds of PR proteins, and their properties has been given here. And coming to the toxins and the classification of toxins in plant pathology, mainly we can observe three kinds of toxin: phytotoxins, vivotoxins, and pathotoxins. So rather than explaining all those things, let me give an examples. The examples of phytotoxins are alternaric acid, lycomarasmin, and cochleobolin or cochleobolin. All are comes under phytotoxins. Okay. When it comes to vivotoxins, fusaric acid, pyrrolidine and lycomarasmin, all are vivotoxins. So the concept of vivotoxin or the term vivotoxin was coined by Diamond and Wagner in 1953. So when it comes to the pathotoxins, pyrrolidine, tentoxin, tabtoxin, victorin, all are the pathotoxins. So among these three toxins, pathotoxins are very important, and they are involved in the major, which plays a major role in disease production. So they will ask, so which of the following toxin is a phytotoxin, or which of the following toxin is a vivotoxin, something like that they will ask. So you guys just remember, so alternaric acid, lycomarasmin and cochleobolin are the examples of phytotoxin. Fusaric acid and pyrrolidine are the examples of vivotoxin. Pyrrolidine, tentoxin and tabtoxins are the examples of pathotoxins. So these are all some host specific toxins. So in toxins, host specific toxins and non-host specific toxins are there. So these are all some host specific toxins like HV toxin or Victorine produced by Helminthosporium victoriae, HC toxin which is produced by Helminthosporium carbonum or Cochleobolus carbonum, HS toxin which is produced by Helminthosporium saccharine or ice pot of sugarcane, HMT toxin that is Helminthosporium maedis resti, ACL toxin Alternaria citri, AL toxin Alternaria lycopersicii, AK toxin Alternaria cucciana, AM toxin Alternaria mali, PC toxin Perniciosa circinata or circinata, PM toxin Philosticta maedis. So these are all the various kinds of host specific toxins and the pathogens produced. So when it comes to this non-host specific toxins, alternaric acid, zinol or alternarial produced by alternarial species, these are all the non-host specific toxins. So fumaric acid, rhizopus species, oxalic acid, aspergillus, uh, taxotomins, streptomyces, syringomycin, syringotoxin, tetrodotoxin, pseudomonas syringae, pathover syringae, fasciotoxin, pseudomonas syringae, pathover fasciolicola, tap toxin or wildfire toxin, pseudomonas syringae, pathover tabasi. So these three are very very important. They will ask, so syringomycin is toxin is produced by the pathogen. Pseudomonas syringae, pathover syringe, something like that they will ask. So syringomycin, syringotoxin and tetrodotoxin which are produced by Pseudomonas syringae, pathover syringae. When it comes to fasciolitoxin which is produced by Pseudomonas syringae, pathover fasciolicola. So when it comes to tap toxin, so this tap toxin is otherwise called as wildfire toxin which is produced by Pseudomonas syringae, pathover tabasi. 10 toxin alternate tenus, Ophiobolin, Cochiliobolus species are Elentosporium varizae, Pyricularia varizae or Pyricularia grisea, Amylovorin, Erwinia amylovora, Cercospora species, Colotrotricum, so Colotricum, Diaphorthin, Endothea parasitica, Fusicoccum amylacti, Helminthosporium sativum, Fusaric acid and lycomaraspin which is produced by Fusarium oxysporum, pharma species Lycopersicii, and Ceratolmin, Ophiostoma ulmi which causes Dutch elm disease. So Dutch elm disease is caused by Ophiostoma ulmi or Ophiostoma nova ulmi, and Fusicoccum amylacti. So these are all various kinds of non-host specific toxins and the pathogens produced. So coming to some important toxins and their mode of actions. Before that, we have seen some fungicides in applied plant pathology sections. We have seen some fungicides and their mode of actions. Now we will see some important toxins and their mode of action. So wildfire toxin or tap toxin, which is produced by Pseudomonas syringae, pathover tabasi, which inhibits the glutamine synthetase activity. Victorin, Helminthosporium victoriae, affects and damages the plasma membrane permeability. Tage toxin, which is produced by Pseudomonas syringae, pathover tagetis, inhibits chloroplast RNA polymerases. Fascioli toxin, Pseudomonas syringae, pathover fascioli, affects the membrane permeability and pyrimidine biosynthesis and host enzyme lipid synthesis. Tab toxin, it's a very important, which is produced by Pseudomonas syringae, pathover syringae. This tab toxin is otherwise called as wildfire toxin. I think I have placed this 2 times. Wildfire toxin or tab toxin, Pseudomonas syringae, pathover syringae. The mode of action is inhibits glutamine synthetase. Pyricularin and syringomycin, which is produced by Pyricularia varizae, whereas syringomycin is produced by Pseudomonas syringae, pathover syringae. So the mode of action of this pyricularin and syringomycin is inhibits membrane permeability. 10 toxin alternate tenus, so reduction of chlorophyll content inhibits the photophosphorylation, causes stomatal closure. Lycomarasmin, Fusarium oxysporum, pharma species Lycopersicii, and Cercospora zeamidis, Amylovorin, Erwinia amylovora, Ophiobolinae, Helminthosporium varizae, and their mode of action has been given here. We may expect maybe sometimes. So let's we'll see something about a chestnut blight. So this chestnut blight, otherwise called as chestnut canker, which is caused by Cryphonecteria parasitica, previously called as Endothea parasitica. So it's a very important. So chestnut blight is caused by Cryphonecteria parasitica. They will also ask like this. So what is the old name of chestnut blight? Endothea parasitica. Now it is called as Cryphonecteria or Cryphonectria parasitica. So the symptoms includes cankers, dieback and death of above ground parts. Actually, this was a very serious disease in United States of America in the period between 1904 to 1944. And almost in the 40 years, just more than 4 billion American chestnut population has been wiped out only because of this chestnut disease. And hypovirulence, so this phenomenon is actually associated with the chestnut blight, because this is hypovirulence is the best mechanism for the management of chestnut blight. So this mechanism actually includes presence of or double stranded RNA. By using this double stranded RNA, we can reduce the virulence of the pathogens, or we can reduce the virulence of the pathogen or pathogenic strains, that is Cryphonecteria parasitica, Endothea parasitica, by using this double-stranded RNA molecule, double-stranded RNA in the viruses. So if we get infected, it can be easily reduce the virulence of the pathogen or fungal infection, so that we can escape the disease or we can minimize the risks. So Dutch elm disease is actually caused by Ophiostoma vulmi or Ophiostoma nova vulmi. So Dutch elm disease is actually transmitted by bark elm beetle, that is Scoletus strawberry. Dutch elm disease is transmitted by bark elm beetle. It's a very important. Oak wilt is caused by Bridgiella fagacearum. It's previously called as Ceratocystis fagacearum. Now the name changed to Bridgiella fagacearum. So oak wilt is actually transmitted by Nutilid beetle, which is belonging to the family Nutilidaceae, and also spread through spores. Here I'm coming once again. Dutch elm disease is transmitted by Ophiostoma vulmi or Ophiostoma nova vulmi, and it is transmitted by bark elm beetle. When it comes to oak wilt, it is caused by Bridgiella fagacearum. The previous name is Ceratocystis fagacearum, and oak wilt is transmitted by a beetle species, that is Nutulid beetle. So butternut canker is caused by Cirrhococcus clavigiganti jugalendacearum. It is otherwise called as Ophiognomonia clavigiganti jugalendacearum. So in sometimes we may expect such kind of questions also. So butternut canker is caused by Cirrhococcus clavigiganti jugalendacearum or Ophiognomonia clavigiganti jugalendacearum. Cypress canker is caused by Ceridium cardinale or Ceridium unicormi or Ceridium cupresi. So cyclosporin is a drug which is actually used in the surgery, that is derived from Toliposporium infloretum, which is the anamorphic stage of Cordyceps subsilicis. So now we'll see about resistant genes, or like R genes in the plants. So actually the concept of this resistant genes or cloned genes were first the first resistant gene was isolated from HM1, that is Johal, Briggs and Walton in the year of 1992. So the first cloned resistant gene is HM1 from maize. It is also very important. What is the first cloned resistant gene? HM1, isolated from maize. Who discovered this first cloned resistant gene or R gene? Johals or Briggs or Briggs and Walton. Sometimes they will also ask Johal, Briggs and Walton. Okay, so this actually detoxifies Helminthosporium carborum, that is H3 toxin, that is a host specific toxin that I think we already seen. So that is a fungal toxin, Cochleobolus carbonum. Now we will see some of the R genes or resistant gene. The first resistant gene is HM1, that is isolated from maize or Helminthosporium carbonum. Tobacco TMV resistant gene is N1, flaxseed rust resistant gene is L6, xanthomonas varaceae XA21, CF2, CF4, CF5, CF9 that confers the resistance genes are resistant. These are the resistant genes that confer the resistance against Cladosporium fulvum, RAS2, RAS4, RAS5 and RAS7 that carries the A virulence genes of 2, 4, 5, 9 respectively. So coming to these are all the major classes of resistant proteins. NBS, LRR, TAR, NBS, LRR, CC, LRR, TRD. These are all the various kinds of resistant gene along with their examples. So LRR means leucine rich repeat. NBS means nucleotide binding site. TAR means toll interleukin 1 receptor. CC means coiled coil. TRD means transmembrane domain. PEST means amino acid domain. ECS means endocytosis signaling cell domain. NLS means nuclear localized signal. WRKY means amino acid domain. So these are all the various full-fledged terminologies about something that we had mentioned in here in about resistant genes or proteins. So these are all the major classes of cloned plant resistant genes that I will tell you. HM1 that we already discussed, that is the first plant resistant gene isolated from maize, Helminthosporium maedis race 1, and PTO tomato, Pseudomonas syringae pathovar tomato, PBS1 Arabidopsis, Pseudomonas syringae pathovar fasciolicola, PBS2 also same, and RP1 Arabidopsis, N tobacco, L flax rust, that is a pathogen tobacco mosaic virus for N. L6 is the resistant confer resistance against Melanospora arini. So these are all the various kinds of genes that has actually, like major classes of genes that are identified in the plants that confer resistant to the pathogens that I have given here, and their isolated years are given here. So coming to the avirulence genes, so now we will see some avirulent genes. Rice AVR peta isolated from, that is the pathogen is Magnoporthe griseae. Matching R gene is peta PATA. Tomato AVR PTO Pseudomonas syringae pathovar tomato PRF. Tomato AVR RPP8 Melodogon incognita or Macrosiphium U4BA MI. Potato code protein potato virus X and RX. Potato elicitin and AVRD Pedobdor infestans PTO. Tobacco replicase tobacco mosaic virus, so sorry, tobacco mosaic virus actually TMV not PVX, and matching R gene is N. So coming to these are all the various AVR genes. The Cladosporium fulvum AVR2 contains 78 amino acids. CFAVR4 contains 135 amino acids. CFAVR9 contains 63 amino acids. So when it comes to the Magnoporthe AVR gene is AVR-PTA 223 amino acids. Pseudomonas syringae pathovar tomato AVR-D, AVR-PTO, Pseudomonas syringae pathovar glycemia AVR capital D, AVR-D. Xanthomonas campestris pathovar vesicatoria AVR-BS2, Xanthomonas citri PTHA, Xanthomonas campestris pathovar malvacearum AVR-B6. These are all the various kinds of AVR genes. So coming to the RNA interference or RNAi, so this these are all various other names are there that I will tell you. So RNA interference or post transcriptional gene silencing, that is the PTGS, post transcriptional gene silencing or RNA interference, otherwise called as co-suppression or quelling. So actually by using the double stranded RNA molecule we can inhibit the expression of genes, so that automatically regulates the production of proteins, that I will tell you. So this entire RNA interference mechanism relies on a transcription and translation mechanism. So we first of all we need to know what is transcription and translation mechanism. So if you see the central dogma of life, in transcription process from DNA, RNA is formed. RNA, where DNA is converted, the message is converted into RNA or messenger RNA. The process of conversion of DNA to mRNA or messenger RNA is called transcription. So DNA to mRNA or RNA is called transcription. So RNA to proteins. So RNAs need to translate. So the information that is copied from DNA is need to translate so that the proteins are formed. So once the proteins are formed, the replication will happen. So the process of conversion of DNA to RNA is called transcription. From RNA to protein is called translation. So this RNA interference mechanism simply silences the translation mechanism or inhibits the expression of genes so that the proteins won't be formed. So here the mechanism was first described or discovered by Andrew Fire and Craig Mello. This question was also asked many times. So Andrew Fire and Craig Mello first discovered the mechanism in a nematode called Keynote elegans. It is also very important. So the questions will be asked like this. So who discovered the mechanism of RNA interference? Andrew Fire and Craig Mello. So Andrew Fire and Craig Mello discovered the RNA interference mechanism on which nematode? Keynote elegans. Or which of the following organism where RNA interference was discovered? Nematode, fungi, bacteria, virus, something like that they will ask. So the RNA interference mechanism was first discovered by Andrew Fire and Craig Mello in a nematode called Keynorhabditis or Keynorhabditis elegans in 1998. For that they got Nobel Prize in 2006. So Hamilton and Balcombe was the first to report the post-transcriptional gene silencing in plants. So if this RNA interference is in plants, it is called post-transcriptional gene silencing. If we observed in a fungi or maybe some other organism, quelling, I think so. So now we'll see meristem tip culture. So now we are going to see this biotechnology in plant disease management. So meristem tip culture was first identified by Martin and Morel. So by using this meristem tip culture, so like meristem is the continuously dividing cells. So if we develop some like plants or plantlets or tissues or whatever it is, the the virus will be completely free, or maybe virus cannot grow in the meristem cells because of continuous divisions. So by using this meristem tip culture, elimination of viruses can be possible. The viruses like PVA, potato virus A, potato virus X, potato virus Y, potato virus S, potato and potato leaf roll virus, tobacco mosaic virus, cauliflower mosaic virus, cucumber mosaic virus in banana, can be eliminated by using this meristem tip culture. So agrobacterium mediated gene transfer. So let me tell you something, since we are discussing this molecular basis, let's go in deep a little bit. So this TA plasmid, tumor inducing plasmid, right? So which is actually can be observed in a bacteria called agrobacterium tumefaciens, so which normally causes galls or tumors in fruit trees like apples, like all stone fruits. So we'll see in TA plasmid two important components are there: octopine, nopalin. So octopine contains two DNA regions, so TL and TR region. So TL region is a 14 kb, whereas TR region is a 17 kb. So when it comes to nopalin, which contains a tDNA region of 20 kb and is size about 200 kb. The questions will be asked like this. So in octopine, what will be the size of TL region? 14 kb. Since it is a molecular plant pathology, definitely they will ask some deep questions also, maybe for confusing us. So I'm just sharing the knowledge whatever I have. So as I told you in the first slide itself, still like it's a puzzling topics for me. So many topics are still a puzzling aspects for me. So let me share my knowledge. Okay, hope you guys understand. So octopine, two tDNA regions, TL and TR. TL is 14 kb, TR is 7 kb. So they will ask what is the size of nopalin plasmid? 200 kb. Okay, tDNA region is 20 kb, but the total size is of 200 kb. So now we'll see the structure of TA plasmid. So TA plasmid is mainly contains two regions, that is the tDNA and the Vir regions. So this T region contains a T plasmid which is of 23,000 nucleotide pair segments. So this actually carries 13 unknown genes, 13 unknown genes. So like and it also contains like auxins, indole 3-acetic acid, cytokinins, various other kinds of stuff also there. So like this contains when it comes to vir genes, I need to talk something about this vir genes, like vir A, vir G, vir B1, vir C1, vir D1, vir D2, vir D4, vir E1 and vir E2 are the like 9 vir regions which operates or confers virulence to the pathogens. So hybridoma technology and production of monoclonal antibodies, that I will tell you the direct questions what we can expect. Though the technology of production of monoclonal antibodies is called hybridoma technology. It is a technology where monoclonal antibodies are can produce. The hybridoma technology was first developed by Kohler and Milstein in 1975. So for that they got Nobel Prize in 1984 for the physiology and medicine. So but the term hybridoma was caused by Leonard Harzenberg. So they will ask questions like this. So the term hybridoma technology was first developed by, or like coined by, something like that they will ask. Kohler and Milstein in 1975. It is a revolutionary technique, and like it saved millions of lives. So hybridoma technology, and it's a very time consuming, time taking process, and also very expensive. So this hybridoma technology is for monoclonal antibodies production. This technique was developed by Kohler and Milstein in 1975. So we'll see some questions related to this hybridoma technology. So what is the specific medium used for hybridoma technology? Is HAT medium. HAT, HAT. HAT means hypoxanthine aminopterine thiamidine is the specific medium used for the production of monoclonal antibodies. So one thing, this the viruses contains reverse transcriptase enzyme, also known as retroviruses. So the enzyme reverse transcription was independently isolated by Mark Temin and David Baltimore. So actually this is related to virology. Don't consider this, just leave it. So CRISPR-Cas, the most advanced technology, CRISPR-Cas. So before let's start, entering into this, let's see what is CRISPR. CRISPR, clustered regularly interspaced short palindromic repeats. So this is a short palindromic repeats because all the DNA sequence was repeated again and again at the adjoints, and the sequence will the sequence will extends. So CRISPR-Cas is the most advanced technology for genomating. We can edit the genome of our interest, we can edit whatever we want, and it is the most advanced technology. For the discovery of this CRISPR-Cas, Dodana and one more scientist, actually I forgot her name, in 2020 or maybe 21 they got received Nobel Prize. So this technique was actually first reported in Escherichia coli by Ishno in 1987, and trust me, the future is about CRISPR only, because it's a genomating technique. We can edit the genome of our interest, so that we can edit cancer genes, or like we can edit whatever we want, and we can design the baby whatever we want. So like in a like a sci-fi films, we can the future will be possible like a sci-fi films with the help of this CRISPR-Cas. So the difference between the genomating tools, as I told you, that the CRISPR-Cas, not only CRISPR-Cas, other genomating tools are the ZNF, zinc finger nuclease, TALEN, transcription activator like elicitor, and the last one is CRISPR-Cas9, clustered regularly interspaced short palindromic repeats. Among these three techniques, so CRISPR is the most important, and CRISPR is the most advanced technique, and most featured technique when compared to ZNS and TALEN, and the description was given here. Hope you guys can go through. So coming to these are all some list of DNA markers has been given: RFLV, restriction fragment and polymorphism, Botstein, RAPD, random amplified polymorphic DNA, Williams, AFLP, amplified fragment and polymorphism, WOS. So these are all the various kinds of DNA markers and their nomenclature, year of discovery, and the references, in the sense the man who discovered it, and all those things has been given. So this slide also represents the same. Again this slide also represents the same. Again same. Yes, and hope you guys already seen this loop mediated isothermal amplification 2000 Notomi. This technique was developed by Notomi. So actually seed pathology was not there in ARS. So let's see just some common things. So because what I'm going to display you, that can also comes under mycology, and as well as principles of plant disease management, something like that. So hot water treatment, and for various plant pathogens. So black rot of cabbage, Xanthomonas campestris, Pathover campestris, 50 degrees centigrade for 20 minutes or 20 to 30 minutes. Seedling blight of cucumber, Pseudomonas syringae, Pathover lacrimens. So 50 degrees centigrade with 75% relative humidity for 3 days. Downy mildew of palm millet, Sclerospora graminicola, 55 degrees centigrade for 10 minutes. Rice udbata, Ephelis varizae, 5410. Potato phyllodes, Phytoplasma, 5010. Hallow stalk of tobacco, Irvinia keratovora, Pathover kerativora, 50-12 minutes. Black speck of tomato, Pseudomonas syringae, Pathover tomato, 50 to 1 hour. Wheat thundu disease, Anguna triticale, 54-10 minutes. Peanut testa nematode, Aphelingoides arachidis, 60 minutes for 60 degrees centigrade for 5 minutes. After that, soaking for 15 minutes in cool water. Stem nematode, Reitlingus displaceae, 50 degrees centigrade for 1 hour or 48.8 degrees centigrade for 2 hours. Rice white tip nematode, Aphelingoides baceae, 51 to 53 degrees centigrade for 15 minutes. So coming to hot water treatments for sugarcane based only sugarcane diseases: red rot, Phytoderma tucumenensis, 54 degrees centigrade for 8 hours. Whipsmut, Sporosorium cetaminium, before it is called as Istilago cetaminium, I think so. Istilago cetamine, now it's called as Sporosorium cetamine, 54 degrees centigrade to 60 degrees centigrade for 10 minutes. Raton stunting, Lifesonia xylem, subspecies xylem. Now previously it is called as Clavibacter xylem, subspecies xylem. It is also very important. Ratoon stunting is caused by Lifesonia xylei subspecies xylei. So previously called as Clavibacter xylei subspecies xylei. So that can be controlled by 50 degrees centigrade for 3 hours. Wilt Fusarium moniliforme 50 degrees centigrade for 2 hours. And downy mildew Sclerospora secari 54 for 1 hour. Leaf scale Xanthomonas albinians and soaking in cold water for 1 day, and then treating with the cutting of 50 degrees centigrade for 2 to 3 hours. Grassy shoot and white leaf phytoplasma 54 2 hours 54 40 minutes respectively. Sugarcane mosaic virus 52 58 for 20 minutes. Spike virus 52 degrees centigrade for 1 hour. So this is a little bit some important topics about physiological and molecular plant pathology. So there are other topics also there like ETL, PTI, ETI, so many other topics are there. So it will be little bit we can't completely cover. There are a lot to look into this physiological and molecular plant pathology, but I have done my level best. Hope it will be useful for you guys. Stay geeky, stay tuned. We are Team Geeky Researchers.